So thank you so much for the opportunity to present my work and the work of our team to this audience. Obviously, it would have been much better to be in person. I've never been there in Costa Rica, but let's start with this virtual class, and then maybe in the future we have the opportunity um, to collaborate. Again, I'm an assistant professor at Penn and the scientific director of the lymphoma program. And I think what I wanted to share with you today is a little bit about our work on CAR T cell for B cell neoplasm and thinking about the resistant mechanism that affect our patient and how we can take advantage of that knowledge to build new therapies. So these are my disclosures, and what I would like to talk to you today is about CAR T cell therapy for cancer and trying to think a little bit about the mechanism of resistance and how to overcome some of them. And I think this audience doesn't need a long introduction, but for the students and for the people new to the field, CAR T cell therapy includes the redirection of patient T cells using typically lentiviral or retroviral vectors. And now with this transduction, the T cell become CAR T cell, which means they express on the surface a chimeric antigen receptor, which can recognize a target in cancer cell and kill the cancer cells. And the reason why I'm showing you this slide is because I think it's important to think about the mechanism by which they kill tumor cells. And as you can see here, you get the CAR T cell forming a synapse with the leukemic cells, and you can see that rapidly the leukemic cells start to blab and then die by apoptosis and other mechanisms. And so far, several products have been approved by the FDA in the US, in particular Kymriah or tisagenlecleucel. This was developed at Penn by the group of Carjun. And soon after, Yescarta or axicabtagene was approved for lymphoma. And then, of course, both these products have now multiple indication in lymphoma and acute lymphoblastic leukemia. After that, we got other products. As you can see here, Tecartus or brexucabtagene autoleucel for mantle cell lymphoma and adult B-ALL, Breyanzi or lisocabtagene maraleucel for large cell lymphoma, and now both Breyanzi and Yescarta have indication in second line. And most recently, we got two products, idacabtagene and cilta-cel, that you can see here, that are both approved for BCMA positive multiple myeloma. But of course, even if the FDA approved indication, we want to see what are the long-term outcome data to really understand if this therapy is able to cure most of our patients. And so we published this paper last year, and this is based on the pilot single-center clinical trial that Dr. Schuster ran at Penn, studying what is now known as tisagenlecleucel. At that time was called CTL019 in diffuse large B-cell lymphoma and follicular lymphoma predominantly. And what you can see here, that at this very long follow-up, 5 years plus, we have about 30% of patients with large cell lymphoma surviving and without disease at long term. And in follicular lymphoma, it's a little bit higher, it's 40%. So I think that it's become pretty clear that we need to enhance these CAR products, because even though we can cure a subset of them, still the majority of them do not respond to this therapy at long. And if we look at the same product, tisagenlecleucel, and look at two registrational trials, the first two that were ran by Novartis globally, you can see that in pediatric and young adult B-ALL, there is a pretty promising event-free survival curve, and with about 50% of the patients that are without disease at about 2 years. And what you see here in the Juliet trial, this is for the adult diffuse large B-cell lymphoma. As mentioned before, for the single center study, we have about 30% of patients that are without disease at long term. So again, clearly remarkable results for patients that would have had otherwise a very poor prognosis, but at the same time, the majority of patients will fail. Either they don't respond right away, like in the case of lymphoma, or they would relapse at follow-up. So really, my group and my lab in recent years has been really focused in studying the resistant mechanism and trying to develop sort of the next generation of approaches that are really based on understanding what are the mechanisms.
And in this paper, in this review paper that we published last year, we tried to sort of summarize the key mechanism of resistance to CAR T-cell immunotherapy. Really, what are the key factors that can inhibit the interaction between the CAR cell and the tumor cell? And so we divided them in four main categories. I always like to start with the pre-infusion barriers, because really even before CAR infusion, we might have reasons that would affect our ability to use this immunotherapy. For example, patients with low lymphocyte counts are patients that might not be able to collect enough T cells for manufacturing. Unfortunately, some patients, even though they can collect the T cell with diaphoresis, they fail manufacturing. And another issue is that while we wait for production of the CAR T cell, which might take between the 3 weeks and even 2 months sometimes, some patient might progress and we might not be able to control the disease. And lastly, which is a topic that is very important, especially for this audience, different healthcare systems might have, and different geography might really impact the access to this therapy. For example, there are entire states in the US that don't have a CAR T-cell center. And of course, places like Costa Rica, or even I'm originally from Italy, places like Italy and Europe, where there is a government-based healthcare system, might have issues on reimbursing these products that are so expensive. But if we look at the more biological reason for failure, I think there are three main categories, right? And of course, there are overlaps. The CAR dysfunction, the dysfunction of the T cells, is a really important factor that might affect how the T cell work. Exhausted T cells, for example, are known to have lower toxicity, decreased toxicity, and also not be able to persist long term or survive the toxic tumor microenvironment. And likewise, T cells that have been going through differentiation and they are terminally differentiated, again, might not have an optimal anti-tumor effect. Then the tumor microenvironment and in general host factors can play an important role in inhibiting T cell function and supporting tumor growth. The physical barrier between the blood and the tumor site, it's an important one. There are soluble factors like TGF-beta or IL-10 and others. Tregs, tumor-associated macrophages, and myeloid-derived suppressive cell also can inhibit T cell function. And so, and other host factors like tumor burden or the gut microbiome might affect, and we show that T cell function. And lastly, the tumor intrinsic mechanism, otherwise known as factors that are strictly related to the tumor cells. For example, a very common one is antigen loss. We've done a lot of studies in patients that don't have antigen loss but still have defects in apoptosis, or for example, even expression of PD-L1, of factors that can inhibit T cell function. And I would like to start with antigen loss, which has been described initially in ALL, but I will show you some data also in lymphoma.
Antigen loss was something that was unheard before CAR T cell therapy. This was just very rare to see a disease that would lose the antigen to avoid a specific therapy. It's really only when you have a strong selective pressure that you start to see loss of the target. And as you can see here, this is just an example of flow cytometry at baseline, nice double expression of 19 and 22, and at follow-up 9 months after CAR relapse without CD19. And this is seen, for example, up to 75% of patients in the ELIANA trial. This is the registrational trial in ALL. But also in pediatric ALL, it's also pretty frequent in the real world, about 40%. The occurrence of this in lymphoma is less clear. Some data show that it could be about 30% in the axis cell population. And so in this study, we really wanted to understand what was the incidence of antigen negative escape in lymphoma for patients that were treated with tisagenlecleucel, so with a different costimulatory domain for 1BB as compared to CD28. And as you can see here, we took about 26 biopsies, all of them with tumors, as highlighted by the expression of 79A. And interestingly, we started looking at another commonly used target, which is CD20. And as you can see here, about 20% of the patient didn't have CD20 at relapse. So this is something that probably was already present at baseline. We're looking into that, and due to the use of rituximab and therapies targeted to CD20. But look at this number. About 40% of patients after CAR19 didn't show expression of CD19 or very, very low expression by immunohistochemistry. And just to give you an example here, you can see here 3 different patients, all of them with lymphoma in the lymph node. You can see that all of them are characterized by 79A expression, which is a marker for B cells. But see, some of them here, for example, this patient number 2, doesn't have CD20, or patient number 3 doesn't have CD19. And so obviously, when we think about the next generation of therapies, we need to think that previous treatment definitely affect the antigen expression of the disease at relapse. But what are the mechanisms leading to loss of CD19? Well, certainly there is a lot that we need to learn, but a few studies have shown key mechanism of resistance. For example, the mutation of the CD19 gene has been shown in both leukemia and lymphoma. And another interesting phenomenon is, for example, the enrichment of splicing variants that don't carry the epitope of that is recognized by the CAR. These variants have then been shown not even to make it to the surface, and so they not only they don't express the epitope, or this one, for example, doesn't express the transmembrane domain, but they even not express on the surface, and so clearly that leads to failure of CAR19. But there are other type of CD19 negative relapses that really speak for the ability of tumor cells to be plastic, to have plasticity, which meaning that they can, for example, in the case of transdifferentiation, we can see that lymphoid blasts can turn into myeloid blasts under the pressure of CAR19. And that really means that the disease is at the level of the stem cell, and now we see switching of lineage because of this selective pressure. Or even more surprising is this case where we saw that a patient who had mantle cell lymphoma was treated with CAR-19, and at relapse, or at least what was expected to be the lymphoma relapse, was actually a sarcoma. And when this group at Penn looked into the VDJ rearrangement, saw that also the sarcoma cell had the VDJ rearrangement, indicating that really those cells were derived from the initial lymphoma cells. So for many patients, we don't know what's the mechanism. We might not see mutation, we might not see transdifferentiation, and so we're still looking for other mechanisms. And a few years ago, we discovered a new mechanism of pseudo loss of CD19 that we call epitope masking. So what happened in this patient? Well, what we observed for this patient is that unfortunately the patient had a relapse. You can see here, this is a leukemia, a pediatric leukemia with relapse in the bone marrow. These are CD45 dim cells that express, as you can see here, CD10, CD22, so they are B cells and BALL cells. They don't have CD3, as you can see here, these are the CD3 positive. But shockingly, all of them were expressing the CAR on the surface. So this means that the CAR, instead of being only on the T cell, was also expressed in the leukemic cells. And so we tried to understand more about the mechanism of this finding. And what we saw is that, and what we demonstrated is that during manufacturing, a single leukemic cell got transduced with the lentiviral vector. And so what happened is that now you have a CD19 positive leukemic cells that start to express a CAR-19 on the surface. And what that means is that this CAR, we demonstrated, has the potential to bind cis to CD19. And so when we try to stain, by flow cytometry, this blast for CD19, we don't see anything because the epitope is busy being bound to CAR-19. It's the same epitope of the antibody and the CAR. And likewise, our CAR T cells are not able to target this leukemic clone because, again, the epitope is bound to the CAR and cis, as you can see here. And so in a second paper, we thought about, can we take advantage of this aberrant expression of the CAR and make a product, another CAR product that is specific for DCAR? In other words, developing a CAR that instead of recognizing CD19 would recognize the CAR. And so we can use this product to kill CAR positive cells. And so we develop an anti-CAR-19 CAR that binds to the CAR-19 and potently kill the leukemic cells that express the CAR. Obviously, one might speculate that this product in the future could be used as a sort of an antidote to CAR-19 in patients that are very long-term, they still don't have B cells and they're in remission and so on. Obviously, that would be a very difficult choice to be made. And so we discussed about different mechanisms of CD19 negative loss, and I think we also need to think, well, if we understand the mechanism, we still have to treat these patients. And so what are the potential strategies to target CD19 negative escape? Well, again, we are now using all the FDA approved products are single targeting CARs. The first concept that we tested in the clinic is the combination of two populations, 1 and 2. One population, for example, targeting CD19, and a second CAR population targeting CD22. And these are called pool CAR. Another strategy is to express either two CARs or a single CAR that recognize two antigen. And the advantage here, especially for the dual CAR, which we think are more potent than the tandem, they are a single population. So one construct, one single population, and one product. Or, more recently, we can develop 4th-generation CAR, for example, CAR T cell that have a CAR, but they also secrete a bispecific antibody against another antigen. or directly taking one of the approved CAR products and use it in combination with a bispecific antibody. So I will show you some data, some clinical data on the pooled CAR, and describe the trial that include combination of these, just to get a sense what is our approach for these issues. So the first study was presented at ASH last year, led by Dr. Fry at Penn, included patients with adult BALL treated with this combination of humanized CAR-19 plus an improved version of CAR-22 that we reported in Nature Medicine again last year. And this is an interesting approach because, again, you have two populations of CAR T cell, and these are co-infused into the patient, 10% at day 0, 30% at day 1, and 60% at day 2. And this is due to the fact that you want to make sure that if there is toxicity, you can interrupt the infusion of these fractionated doses. And the results are pretty remarkable, as you can see here. For 11 patients that are evaluable at day 28 at the time of this publication, you can see that basically most patients reached a complete response, and the complete response is in all cases is MRD negative. And so far, the vast majority of these patients are continuing to respond. I mentioned another possible approach that I think is promising is to combine CAR T cell with a bispecific antibody. You can see this trial here that is led by Steve Schuster at Penn, where CAR T cell are infused at day 0. At day 30, a bispecific antibody called mosunetuzumab, we also have a cohort for glofitamab, is infused. There is a first cycle, second cycle, then disease restaging. If the patient is in remission, stop treatment. If not, if partial response or stable disease, continue the bispecific, or finally, if the patient unfortunately is progressing, is off study. And obviously here is the concept to target CD20 and CD19. The bispecific is against CD20. But also the idea that the bispecific can enhance CAR T cell function potentially. So we discussed a lot the 19 negative relapses, but I think it's important also to think about the CD19 positive relapses. Usually they happen when the T cells are not very functional, but sometimes we do see T cells that proliferate, they persist, but still the patient relapses. And we sort of set to study this in the lab and then in the clinic. And so to summarize some of the work that was published before, we took two unbiased approaches. The first one was a genome-wide CRISPR-Cas9 screening. And so in this screening that has been published by Dr. Singh in Cancer Discovery, what we show in this study is that if you knock out one gene per leukemic cells, and then you culture this gene-edited leukemic cell with the CAR-19, you can study the gene knockouts that lead to relapse or the gene knockouts that lead to response. And interestingly, in this initial study, what we noticed is that the gene knockouts that led to resistance, so they were enriched at the end of the co-culture, were the genes that were related to pro-apoptotic factors. For example, FAD, BEAD, caspase-8, or TRAD, as you can see here. So these are all pro-apoptotic factors. You knock them out, and obviously the leukemic cells are more resistant to CAR-19. But it's interestingly because, as you can see here, all the top candidates are in the apoptotic pathways, but in particular in the extrinsic pathway, which is the pathway that is triggered by TNF-alpha, Fas ligand, trail ligand, and so on. And just really to prove that this was the right pathway to study, the gene knockouts that were actually lost, meaning that those cells were more prone to be killed by CAR19, were those ones that are actually anti-apoptotic. So if you knock them out, the cell is more prone to undergo apoptosis. So this was the first unbiased approach. The second unbiased approach was a small molecule screening, where we co-culture small molecules with CAR T cell and tumor cells, and we ask the question, which one are the small molecules that cause increase of killing? And interestingly, we got the top hits were again in the same pathways, were in the extrinsic pathway and also BCL2 inhibitors. So we know BCL2 inhibitors, such as venetoclax, they are widely used in the clinic. And for these targets, which are the inhibitor of apoptosis proteins, there is a class of drug called SMAC mimetics or IAPS inhibitors that are being tested in clinical trials, such as birinapant, that specifically target these cells. And so Yonggu Li, who was a postdoc in my lab and is now a professor at Anyang University in Seoul, showed that if you co-culture birinapant, so these are the SMAC mimetics with CAR T cell, you get a nice jump of killing from 20% to 60%. And keep in mind, birinapant alone does nothing. And this is in a leukemic model. But this was also true for example, ovarian cancer, where you can see that the combination of CAR T cell and birinapant in vitro would lead to a very nice anti-tumor effect. We then, of course, move to in vivo because you want to validate your finding in an in vivo model. And what we found is that in mice, immunodeficient mice engrafted with a leukemia cell line, birinapant does improve the anti-tumor effect as compared to CAR T cell alone. But if you look at long term, the CAR T cell with birinapant actually do worse than the CAR T cell alone, as you can see here. And so, of course, we wanted to investigate the mechanism by which we were seeing these results. And what you can see here is that actually, when you check the expansion of the CAR T cell in the peripheral blood, as we do in patients, we did in these mice, you can see that the CAR T cell that were exposed to birinapant have a much lower expansion, likely due to the fact that birinapant is inducing apoptosis in CAR T cells. And so, of course, we were disappointed, and while we still work at those models, we started thinking about BCL2 inhibition. And we tried to model BCL2 inhibition during CAR therapy using 3 different models. A model that is very sensitive to venetoclax, OCLY18, is a DLBCL cell line, then an intermediate sensitivity, MINO, and a resistant model, NAM6. So MINO is a mantle cell lymphoma, NAM6 is a BALL. And so we did a pretty standard animal experiment with NSG mice engrafted subcutaneously with the tumor for OCLY18 and MINO, and IV for NAM6. I'm showing you the OCLY18 model, where CAR T cell after a week were administered, and venetoclax was given every work week day. And the results were pretty remarkable. As you can see here, the combination of CAR19 and venetoclax led to 100% response and good results at follow-up, while CAR19 or UTD with venetoclax or UTD alone didn't really do much. And this was clearly seen in a pretty dramatic advantage in overall survival of these mice. But of course, we know in the clinic, patient can be sensitive or can be more resistant to venetoclax, and sometimes you need to use higher doses, for example, 50 or 100 milligrams of venetoclax per kilo in mice. And when we did these experiments using MINO and NAM6 cell lines, what we saw was actually pretty disappointing. Again, similarly to what we saw in the birinapant experiment, the mice that were receiving the combination were actually doing poorly more poorly as compared to mice just treated with CAR19. You see the red lines here and the red lines here. So again, what was happening in this patient? Well, again, we measured the CAR T cells in the peripheral blood, and again, disappointingly, we see that the CAR T cells that were exposed to venetoclax were dying. And so we sort of started to come up with an hypothesis. We say, can we develop a system where our pro-apoptotic drugs only affects tumor cell, but does not affect the CAR T cell? And of course, in this case, we decided to learn from a very good teacher, which is for resistant, and this is cancer. And so we look into cancer cells from published reports and how they develop resistance to CAR T cell, to venetoclax. And so it's interesting because some of these tumors develop mutation in BCL2, for example, the BCL2F104L, that really makes BCL2 not sensitive to venetoclax. And so we hypothesize if we express this mutation in CAR T cell, we might make them resistant to venetoclax. That's exactly what we did. And interestingly, you can see here that in vitro, the IC50 of venetoclax on CAR T cells gets a lot better if you just overexpress BCL2 wild type, but even a further log better if you express the mutation. And we tested this concept in the clinic, and as you can see here, notably the combination of CAR19 and venetoclax now is synergistic as compared to controls that you can see here. And this is true for anti-tumor effect, but also for overall survival, as you can see here. And we wanted to see if this was relevant in the clinic. In other words, if BCL2 has a role in lymphoma patients that are treated with CAR. And indeed, we saw that BCL2 translocation and gain that typically lead to higher expression of BCL2 protein were associated with very much reduced overall response rate and reduced overall survival. So another reason, another rationale to target BCL2 in these cases. And lastly, we look into patients with mantle cell who received venetoclax as a bridging therapy. So this was between the apheresis and CAR infusion. And what we notice is that patients receiving sort of a priming with venetoclax had a much higher rate of complete response as compared to patients who did not receive venetoclax. Of course, small cohort of patients, about 20 patients, but still a pretty significant signal. And lastly, we noticed that in these models, actually, just the overexpression of BCL2 independently of venetoclax and the wild type BCL2 was leading to improved anti-tumor effect, which is basically a new finding where we saw that BCL2 overexpression led to advantage in tumor control, overall survival. This is Mino. And this is also NUM6, pretty striking difference. And interestingly, this is due to higher expansion and persistence of this CAR T cell, which obviously makes it an interesting model to study in the clinic. So I want to conclude here. I show you the results of CAR T cell and the need for improvement. We talk about the concept of CD19 negative escape and strategies to overcome CD19 negative escape. And then I switch to the apoptotic pathway, showing you the role of apoptosis in cancer resistant to CAR T cells. And I show you that BCL2 inhibition in CAR T cell could be a strategy to overcome resistance. I really would like to thank all of you for your attention, my current lab. Again, thank you to Young-Gu Lee, who led really a lot of projects in the lab, including the venetoclax story, all the collaborators at Penn that you can see here, all the funding agencies, and again, feel free to reach out to me for questions or for potential collaboration. Thank you.